If you could peer into the brains of people with depression, PTSD, and substance use disorder, you might be surprised by what you find. All of these illnesses and many other neuropsychiatric and neurodegenerative diseases are characterized by the atrophy of neurons in key parts of the brain that regulate motivation, fear, reward, and cognition. If you think of these neurons like a tree, the dendrites would be the branches, and the synapses would be the leaves. And in many brain disorders, the leaves fall off and the branches get pruned. Our group has been studying a class of molecules called psychoplastogens that act like miracle-grow for the brain. And because of their ability to regrow these critical neurons, psychoplastogens actually have the ability to heal the brain, not simply treat disease symptoms. In 2018, our group discovered that psychedelics like LSD are really effective psychoplastogens, and this might explain why molecules like psilocin and MDMA are showing really promising results in clinical trials for depression and PTSD. I'm certain that some patients will benefit from first-generation psychoplastogens, but you have to remember that 20% of the population will suffer from a brain disorder at some point in their lifetime. The scale of the problem is enormous, and if we really hope to move the needle, we need to develop medicines that are safe enough that you can take them home and put them in your medicine cabinet. To that end, our group started using chemistry to engineer analogs of psychedelics that lack hallucinogenic and psychostimulant properties, but retain the ability to regrow cortical neurons and heal damaged neurocircuitry. Our academic discoveries at UC Davis led to the founding of Psychedelics Therapeutics, a company focused on using non-hallucinogenic psychoplastogens to address a variety of unmet needs in brain health. So far, we've synthesized hundreds of new molecules, with several demonstrating rapid and sustained therapeutic effects in preclinical models of several neuropsychiatric disorders. We are very excited to see how they perform in the clinic over the coming years. Our work really highlights the power of interdisciplinary science. When you bring together a diverse group of creative chemists, molecular biologists, and neuroscientists who are all passionate about making the world a better place and who aren't afraid to swing for the fences, you can accomplish amazing things together.
Hello, and welcome to today's Plugged In event. I'm Jason Woolman, Associate Vice Chancellor of University Development, and I'm thrilled to be with you all today to discuss the future of mental health care and its intersection with psychedelics. Today, I'm joined by an expert in the field of psychedelics and neurotherapeutics who will dive into how psychedelics are impacting a variety of brain conditions, revealing how we are moving from the stereotype of deadheads to depression meds. So without further ado, let's now hear from Dr. David Olson, Professor of Chemistry, Biochemistry, and Molecular Medicine, and the founding director of the UC Davis Institute for Psychedelics and Neurotherapeutics, who will introduce us to how psychedelics impact the brain. David?
Hi, everyone. Thanks for joining us today. I'm very excited to tell you about our work to try to understand exactly what it is that psychedelics do to the brain and how we use this information to engineer better medicines, medicines that are not only effective, but also safe and scalable, medicines that can be used for a variety of different brain conditions, ranging from neuropsychiatric diseases like depression, PTSD, substance use disorders, schizophrenia, to neurological conditions like stroke and traumatic brain injury, and even neurodegenerative conditions like Alzheimer's disease and Parkinson's disease. But before I do that, I want to tell you a little bit about how the field has treated a condition like depression for the past 30 years. Now, traditional antidepressants have really relied on mitigating disease symptoms through the use of things like selective serotonin reuptake inhibitors in order to try to rectify chemical imbalances in the brain. And while some patients have definitely been helped by these medicines, they have major drawbacks. Number one, they're very slow acting. SSRIs take weeks to months to demonstrate any kind of clinical efficacy. Number two, they require chronic administration, daily administration. And this chronic daily administration leads to a whole host of side effects
like weight gain, sexual dysfunction, side effects that are so severe that many patients simply stop taking their medicines. And number three, about a third of all patients will never respond to these types of treatments. So clearly, new therapeutic approaches are required. Now, in contrast to what you might have heard, we've known for decades that diseases like depression are not caused by chemical imbalances in the brain. They are diseases of neural circuits. And so our approach has been a little different. Our approach has been to identify small molecule drugs that can repair damaged neural circuits. And then by doing so, we can create medicines that can produce rapid therapeutic effects and sustained therapeutic effects after just one or a couple of administrations. And because many brain conditions share damaged neural circuitry, they can work across disease indications. So we call these medicines psychoplastogens. And in our hunt to identify psychoplastogens, we became very interested in this class of molecules. These are the classic serotonergic psychedelics and related compounds, molecules like LSD, which is associated with the counterculture of the 1960s, psilocin, which comes from magic mushrooms, and MDMA, which has been associated with rave culture. Now, there's a long clinical history of using these compounds to treat conditions like depression, substance use disorders, post-traumatic stress disorder. And more recently, there's been phase 2 and phase 3 clinical trials demonstrating some pretty impressive efficacy. Now, the reason that our group was so excited about these compounds is because they had the exact types of properties that we were looking for. They work rapidly within 24 hours, and then just one or a few doses can produce effects that last for weeks to months. And that is really, really quite remarkable. And then finally, the third thing that was very interesting about these compounds is that they tend to work across disease indications. There's evidence that they might be effective for depression, PTSD, substance use disorders, et cetera, et cetera. When we started this research, what was really lacking was a mechanistic understanding of how these compounds could produce these rapid and sustained therapeutic effects across disease indications. And what we discovered was that psychedelics do something called promoting structural neuroplasticity in the brain. And that is exactly what it sounds like. It is the ability of a neuron to change its structure, to change its morphology. And to understand why that might be important, you need to know something about the structure of neurons in a part of the brain called the prefrontal cortex, which sits right about here. The prefrontal cortex is the so-called master control center of the brain, the executive center. And that's because it talks to a whole bunch of other brain regions that regulate mood, motivation, fear, reward, memory, cognition. And so you can imagine that if you have dysfunction of the prefrontal cortex, you'll end up getting all of the symptoms that we commonly associate with diseases like depression and Alzheimer's disease. And so in a healthy brain, a neuron in the prefrontal cortex looks a lot like a tree. In fact, neuroscientists will often use the term dendritic arbors because the dendrites of neurons look a lot like the branches of a tree. And if you look on one of these dendrites, one of these branches, and you zoom in, you'll see these little tiny nubs. These are called dendritic spines, and they are the sites of excitatory synapses in the brain, the way that neurons communicate with each other. And these dendritic spines would be analogous to the leaves of the tree. And what happens in many neuropsychiatric and neurodegenerative conditions is it looks like winter. The arborist has come in, they've pruned back all of the branches, all of the leaves have fallen from the tree. And what this ultimately does is it impairs communication in the brain. The prefrontal cortex can no longer communicate effectively with those other brain regions, and that leads to changes in mood, motivation, memory, et cetera, et cetera. And so one of the first experiments that we ever did is we just took neurons in a dish and we treated them with psychedelic compounds. And so here I'm showing you tracings of actual neurons that were treated with either the vehicle control, which is in the middle, or the psychedelics dimethyltryptamine on the left and LSD on the right. And you don't have to do any kind of fancy data analysis to see the effect here. You can just visually appreciate that the psychedelic treated neurons have substantially more complex dendritic arbors, demonstrating that they have this ability to promote the growth of these really critical neurons in the brain. Now, we published this report way back in 2018. It made the cover of Cell Reports. And this was a time when the media started becoming very excited about the possibility of using psychedelics to treat mental illness. And so if you've ever picked up a copy of the Wall Street Journal or the New York Times, or heard a story on NPR about how psychedelics promote neuroplasticity, it came from this really pioneering study conducted right here at UC Davis. Every single author on this paper is from UC Davis. And so I'm very, very proud that UC Davis was really leading the field in understanding how psychedelics promote neuroplasticity and might actually heal damaged neural circuits. Now, if you're wondering, will these types of psychoplastogenic medicines ever make it into the hands of patients? Well, the good news is they already have. And so about a year after we published that paper in Cell Reports, the FDA approved this molecule here on the left, ketamine, for treatment-resistant depression. Ketamine is a very old molecule. It's been used in medicine as an anesthetic for a long, long time. It does have some dissociative properties associated with it, but there's been a ton of mechanistic work that has demonstrated that ketamine is likely producing its rapid and sustained antidepressant effects by promoting the growth of those really critical neurons in the prefrontal cortex that I told you about. So here you can see that chronic stress actually leads to the atrophy of those neurons, the loss of those dendritic spines or those leaf-like structures on the branches, and that results in synaptic deficits. But what is really remarkable is that a single dose of ketamine within 24 hours can rapidly regrow those lost dendritic spines and reestablish synaptic connectivity in the cortex. And so this was really, really paradigm shifting for the field. The introduction of ketamine to neuropsychiatry was probably the first mechanistically distinct medicine we've had in the past 30 years. And so our goal has really been to thoroughly understand how ketamine and psychedelics are producing this effect on cortical neurons so that we can engineer optimized therapeutics. It's really important to understand that while ketamine and psychedelics might have therapeutic properties, they were never intended to be optimized CNS therapeutics. And so we've been spending a lot of time trying to understand the basic mechanisms by which psychedelics can promote cortical neuron growth. And we've been trying to figure out what is the target that is really important. And that led us to study this particular serotonin receptor called the serotonin or 5-HT2A receptor. It's well known that the serotonin 2A receptor mediates the hallucinogenic effects of psychedelics. And we did a bunch of pharmacological studies and using some genetic knockouts, we were able to demonstrate that it is also essential for the neuroplasticity promoting properties of psychedelics. Now, this is a receptor that in most cell types exists on the cell surface. And so here I'm showing you in kind of like a non-neuronal cell where the serotonin 2A receptor is expressed on the plasma membrane, on the cell surface. But what we found was that in those critical cortical neurons that I told you about, it exists primarily on the inside of cells. And that was a really, really shocking discovery, because these type of receptors normally exist on the cell surface. But in this case, it really exists on the inside of the cell. And what that told us was that serotonin in psychedelics might produce different effects, even though they both are activators of this particular receptor. And the reason that's the case is that serotonin is too polar, too water soluble, not greasy enough to actually be able to physically enter the cell. Psychedelics have this property that they can very easily cross the membrane of a cell and go inside and come back outside, but serotonin cannot. It can basically just stay on the periphery of the cell. And so that explained for the first time why serotonin does not promote cortical neuron growth or structural neuroplasticity in the same way that a psychedelic can. And so we published this work in 2023 in Science. And ever since then, we've been continuing to extend our understanding of how psychedelics impact the structure and the function of the brain. And we've been using these mechanistic insights in order to develop optimized therapeutics. And so one thing that is immediately obvious about psychedelics is that they produce very profound hallucinogenic effects that limit their clinical scalability. Now, I am a firm believer that these first generation psychedelic compounds like psilocybin might be useful for treating certain patients. Having that type of profound mystical type experience might be very beneficial to particular patient populations. But what you have to realize is that in order to be administered safely and effectively, they have to be given under the supervision of usually multiple clinicians, usually over multiple sessions, a preparatory session, a drug session, and then an integration session. And that just leads to drastic increases in the costs and the complexity of the treatment, making it inaccessible for many people. It's very unclear at this point if insurance companies will pay for this. And our goal really has been to democratize access to these types of medicines that can heal damaged neural circuits. And so when we were thinking about how we would approach this problem, we realized that maybe we could separate the hallucinogenic effects of psychedelics from their beneficial effects on neuroplasticity. And if we could create a medicine that was safe enough that people could just pick it up at their local pharmacy, bring it home, put it in their medicine cabinet, and take it as prescribed by a doctor. And that would drastically reduce the cost, which would drastically increase the number of people that could benefit from it. And not only the cost issue, we would solve some of the comorbidity issues. So for instance, psychedelics, because they produce this pretty profound effect on subjective experience, are contraindicated for people with certain neuropsychiatric diseases like schizophrenia, or even a family history of psychosis. And you have to remember that neuropsychiatric diseases are highly heritable. And so if you're going to receive psychedelic-assisted psychotherapy to treat your depression, there is a good chance that someone in your family has had a psychotic illness, and it might preclude you from partaking in psychedelic-assisted psychotherapy. In addition, psychedelics can have some pretty profound effects on cardiovascular function. So if you have a bad heart or high blood pressure, then you also might be precluded. In addition to people who put their hand up and they say, I want to participate in psychedelic-assisted psychotherapy, but can't because of these co-occurring conditions, there's a whole group of people who simply don't want a hallucinogenic experience, either for personal or religious reasons. And so by decoupling the hallucinogenic effects from the beneficial effects on neuroplasticity, we hope to reach a broader patient population. And so we published the first example of a non-hallucinogenic psychedelic analog with therapeutic potential back in Nature in late 2020, early 2021. And since then, there have been several other academic groups that have published on similar compounds, and about at least 20 companies that have started to develop these non-hallucinogenic psychoplastogens. And so I want to show you data on one of our non-hallucinogenic psychoplastogens that we call tabernantolog, or TBG. And the way this experiment works is we have these mice that have their neurons labeled with a fluorescent protein so that we can visualize them. Essentially, they're like glow-in-the-dark neurons. And we can use a microscope to look into their brains. We can look at a section of dendrite. We can then give TBG, come back 24 hours later, after the drug has been completely cleared from the body, and we can look at the exact same section of dendrite. And what that allows us to quantify is how many of these dendritic spines are formed, like this one here in blue, and how many of them are eliminated, like this one here in red. And what we find is that TBG does not really impact spine elimination, but it does promote spine growth, and it does so to a comparable extent as this hallucinogenic drug here, DOI. So now that we knew that TBG could promote structural neuroplasticity in vivo, we wanted to look at its acute subjective effects. And we can do some behavioral assays to demonstrate that it's likely to not be hallucinogenic, but we can also just directly look at neural activity. And so what I'm showing you here is on the left, this is just the baseline neural activity of an animal before it is given either a dose of the psychedelic 5-methoxy-DMT or a dose of TBG. That's the blue line where they get the drug. You can see that immediately after they are administered the psychedelic, you get this spiking pattern in the brain. You don't see that spiking pattern with the non-hallucinogenic variant. And then we can use particular markers of neural activity and look across the entire brain. So what I'm showing you here is the entire brain kind of rotating around, and these colors are just indicating various brain regions that have been activated following the administration of the psychedelic on the left, and those regions that have been activated following the non-hallucinogenic compound on the right. And you can see that they are very, very different. Again, emphasizing that we can produce compounds that are structurally related to psychedelics, but produce very different acute subjective effects, but produce the same long-lasting effects on neuroplasticity and long-lasting therapeutic behavioral effects. And so TBG was very important because it was the proof of principle. It was the first demonstration that it was possible to do this. But we want to do drug discovery. We want to continue to optimize and iterate on these compounds, and more importantly, to tune them to specific disease indications, to tune them to depression, to tune them to Alzheimer's disease, to tune them to substance use disorders. And in order to do that, we need to be able to test thousands and thousands of compounds rapidly. So that means we need to develop new assays, new tests of neuroplasticity. And we've been working on this for a very long time. And what we found is that we can take cultured neurons in a dish, and we can treat them with psychoplastogens and measure various changes in their morphology and in their structure. And so here's an example of just a fluorescence microscopy image showing the various structural features of this particular neuron. So that has been really useful for establishing the efficacy of these compounds and their effects on neuroplasticity. But we needed a way to determine if a compound was likely to be hallucinogenic or not. And when we started this research, there were no tests, no in vitro tests of hallucinogenic potential. All of the tests that were available would basically lump psychedelics and non-hallucinogenic psychoplastogens in the exact same category. They couldn't really differentiate between those types of compounds. And we knew that the serotonin 2A receptor was the critical receptor that led to both the hallucinogenic effects of the drugs and the neuroplasticity promoting effects of the drugs. But these traditional assays we're looking very far downstream of that receptor activation. If you think of receptor activation like a set of dominoes, when the drug binds to the receptor, that's knocking over the very first domino. And then dominoes continue to fall until you get very far down the line. And you can have a couple of different domino streams converging to result in the ultimate last domino falling. What these traditional tests were doing was measuring kind of the last domino. And as a result, the hallucinogenic and the non-hallucinogenic compounds looked very similar. We decided to look at the very first domino. What if we could develop a test that would directly probe the conformation of the receptor? And we reasoned that there must be a difference between how psychedelics are binding to the receptor and how non-hallucinogenic psychoplastogens are binding. So the assay we developed was quite simple. We simply took the serotonin 2A receptor and we fused on a protein that lights up in response to certain conformations of the receptor. So when a compound binds to the receptor, it causes a conformational change, and that turns on the fluorescence or it turns it off. And what we found was that hallucinogenic compounds like LSD turn on the receptor very, very well, but these non-hallucinogenic psychoplastogens like glyceride, they do not turn it on. And you don't have to be a chemist to appreciate how similar these two molecules are. The only difference is basically this one position and this one atom. And LSD obviously has very profound subjective effects. Glyceride is a non-hallucinogenic compound that at one time had been used to treat Parkinson's disease. So now we have this cellular assay that allows us to screen thousands and thousands of compounds to predict their hallucinogenic potential. We published this work in Cell in 2021, and it was voted the #2 biomedical discovery of the entire year by Stat Madness. And so we've been using this biosensor that we call Psychlite to predict hallucinogenic potential in combination with our neuroplasticity assays to do drug discovery. And what that has allowed us to do is to take every single major psychedelic scaffold, ergolines like LSD, amphetamines like MDMA, iboga compounds like ibogaine, tryptamines like DMT, and just tweak their chemical structures just a little bit, just rearrange a few atoms to make them better versions of themselves, to make them safer. Every one of these psychedelics has their own unique challenges associated with them. Obviously, we've been talking about the profound subjective effects of compounds like LSD and DMT that limit their clinical scalability, but other compounds have other issues. For instance, MDMA is a psychostimulant, which can lead to abuse liability. Ibogaine binds to an ion channel in the heart that can cause cardiac arrhythmias, and several people have actually died when they took ibogaine to treat their substance use disorders. So by tweaking their chemical structures, we've solved all those issues. We make compounds that are not only non-hallucinogenic, but they're not psychostimulants, they're not addictive, and they don't have any cardiotoxicity risk. So we're hoping that they're going to have much broader therapeutic applicability. Now, because these new compounds are so much safer than psychedelics, we think that they can start to be used in conditions where psychedelics cannot. And one perfect example is schizophrenia. Now, schizophrenia is a disease that's characterized by positive symptoms like hallucinations, but it's also characterized by negative symptoms like depression and cognitive symptoms. And we have really good drugs for addressing the positive symptoms of schizophrenia, but we have nothing to address the cognitive and the negative symptoms of the disease. And we've known for many, many years that the cognitive and negative symptoms of the disease are probably a result of dendritic atrophy in the prefrontal cortex. And so here, this is some imaging from post-mortem brains from patients with schizophrenia. And you can see that compared to the control, the patient with schizophrenia has atrophy of their neurons in the prefrontal cortex, a loss of those dendritic spines. So it would be really advantageous to give patients with schizophrenia psychoplastogens to regrow those lost dendritic spines, but you don't want to give a hallucinogenic drug to a patient with schizophrenia. So we're hoping that these non-hallucinogenic psychoplastogens could be useful in conditions like schizophrenia and ultimately in conditions like Alzheimer's disease. Dementia is another condition where you are not likely to want to give a psychedelic drug because it can induce very profound anxiogenic effects. And if a patient doesn't fully understand what's going on and aren't prepared for that effects, that can be pretty devastating. So for this part of the talk, I just wanted to briefly summarize and tell you that in a healthy brain, we discussed how neurons in the prefrontal cortex have many dendritic branches and many of these little tiny nubs called dendritic spines, and that in many brain disorders, there is physical atrophy of these neurons, and that psychoplastogens, be they hallucinogenic drugs like psychedelics or these non-hallucinogenic psychoplastogens that we've been developing, they're very good at promoting the growth of these neurons to actually physically heal this damaged neural circuitry to produce rapid and sustained therapeutic effects across a number of indications. And so what has this research done for UC Davis? Well, in the past 5 years, I would say that UC Davis research has been featured in the popular press hundreds of times. Every major outlet has covered our research, including things like NPR, the Wall Street Journal, The Economist, The Guardian, BBC. And here I'm showing you an article from the New York Times. It was on the cover of the Sunday opinion page, where they specifically focused on our approach to developing these non-hallucinogenic psychoplastogens. And because we were really leading the field in this basic mechanistic understanding of how psychedelics impact the brain and using that to develop new molecules, new medicines, we decided to found the Institute for Psychedelics and Neurotherapeutics. And this was really a historic moment. We founded the Institute back in 2022. It was a $5 million investment from the Dean of the College of Letters and Science and the Dean of the School of Medicine, along with the Provost and the Vice Chancellor for Research. And I want to emphasize that there are many groups all across the world at universities that study psychedelics. But this was the very first where a major university actually put money into this area, because we can see the potential to really impact people and make a huge difference in the world. And so this $5 million investment is quite consequential for the field. Now, who is the IPN? Well, right now, we are over 50 principal investigators spread across the UC Davis main campus and the School of Medicine in Sacramento, and we span over 30 different departments and centers. And we have a very simple mission. It's a two-part mission. The first is to advance our understanding of the basic neurobiology of psychedelics, and then use that information to develop safe and effective therapeutics, new treatments. And the reason we think that UC Davis is uniquely positioned to tackle this really important goal is because we have a really incredible chemistry and neuroscience community on the campus. Now, here I'm highlighting several of the groups that make up our neuroscience consortium. We have one of the very few primate centers in the country. We have groups like the Mind Institute, which focuses on neurodevelopmental conditions. We have things like the Alzheimer's Disease Research Center. All of these different groups that are very focused in their specific area of neuroscience. And what we are really proposing is that the IPN is going to serve as the drug discovery center for all of these different institutes across campus. And so I want to really highlight how the IPN is different from other groups across the world studying psychedelics. So the very first groups to form around psychedelic science were really at Johns Hopkins and Imperial College in London. But there's other groups too, including those at Harvard and even some of our sister schools like UCSF, UC Berkeley, and UC San Diego. And while all of these groups do some level of clinical work on psychedelics and related molecules, only a few of them actually study preclinical models to understand mechanistically what psychedelics are doing to the brain. And only UC Davis is taking those clinical and preclinical mechanistic insights and translating them into novel chemical matter that can be medicines that pharma companies can develop to help patients. And so this is really how we imagine the IPN working. First, we have to raise funding, and we can do this in a variety of mechanisms, one via private philanthropy, two via grants, or via sponsored research agreements with industrial partners. We then work with all of our different partners across campus in the various neuroscience groups like the Alzheimer's Disease Center or the Mind Institute. And then we can create something new. We can create novel, optimized therapeutics, and then we can license these to large pharma or small biotech to start to bring them to the clinic, or we can spin out our own companies. And so I'm going to show you one example of a spin out company. This is a company that I co-founded called Decoupling Therapeutics. The name Delix comes from psychedelics without the psyche. And they're really focused on developing these non-hallucinogenic psychoplastogens for treating a wide variety of brain conditions. Delix has been relatively successful so far. They've raised over $100 million in venture capital in a Series A. And they've taken one UC Davis compound through phase 1 clinical trials. And I'm going to show you a little bit of that data in a second. They have another UC Davis compound that have completed IND enabling studies and is ready to go into the clinic. And there's many, many other UC Davis compounds in the pipeline ready to go into other disorders. Now, Delix has won a number of awards, including being named one of the Fierce 15 by Fierce Biotech, one of the top academic spinout companies from Nature Biotechnology. And it's just really nice to see a UC Davis spinout company highlighted next to a pharmaceutical giant like Pfizer. So now I want to tell you about one of these compounds that started with an idea here at UC Davis. It progressed through preclinical trials, mechanistic insight, optimization. It was then licensed to the company, who then developed it further, brought it to the clinic, and we tested it in people. Now, all of our preclinical data suggested it was likely to be non-hallucinogenic, but you never really know until you give it to a person. And what I'm showing you here are 16 different questions that you can ask a patient to determine if a compound produces any dissociative or hallucinogenic effects. Now, these different colored lines in the middle, these are different doses of the compound. The compound is called DLX1. This time period right here is when you give the drug. So for these two data points on the left, this is before the drug was given, and the data points on the right, that's after the drug is given. And this green bar that surrounds these things, that's basically a normal response. This is if you have absolutely no hallucinogenic or dissociative effects, you would expect to be within that green bar. And you can see that for every single one of these questions, at every single one of these doses, the compound is non-hallucinogenic. So we did not create just a weak, psychedelic compound that if you go to a very high dose, you start to get hallucinogenic effects. No, we created a compound that cannot produce hallucinogenic effects at any doses because it doesn't engage the same mechanisms to produce hallucinations that psychedelics does and that psychedelics do. But it still might engage the same plasticity mechanism. So this is a phase 1 study in healthy volunteers. This is not a patient population, but we wanted to get an idea of whether or not the drug was impacting these plasticity mechanisms. And so we relied on something called quantitative EEG. And so the way EEG works is you can basically hook up a whole bunch of electrodes to the scalp of a patient. And so here you can see you're looking down on the patient's head. This is their nose, there's their ears, and you have all these electrodes. And then you can give the drug and you can look for changes in particular frequencies. And in this particular case, I'm showing you changes in something called theta power. And it's well known that if you give a psychoplastogen like ketamine, you'll get an increase in theta power. And it's widely believed that this increase in theta power is indicative of these neuroplasticity changes that we've been talking about. And so in this case, when they gave DLX1 to these healthy individuals, you can see that we're going from placebo on the left, and we're going to increasing doses of the compound up to 120 milligrams. And then going down the side, this is the time. So this is 125 minutes before dosing, 71 minutes before dosing. This is 63 minutes after dosing. So you can see that there's really no effect prior to dosing. And as you give the compound, you see this very, very large increase in theta power that is very reminiscent of what you would see when you give something like ketamine, and that it fades over time. So this is really encouraging, suggesting that we didn't make the world's most expensive placebo. We made something that is non-hallucinogenic, but might be causing the type of neuroplasticity that we want to see in phase 2 proof of concept studies. Now, I want to spend the last couple of minutes describing how our work might benefit UC Davis in the long term and might lead to kind of a catalytic cycle of discovery. So I'm going to tell you a story about a medicinal chemist at Northwestern. His name is Rick Silverman. Now, he developed a compound called pregabalin. You might more commonly know of it as Lyrica. Lyrica is a drug that has been used to treat a whole bunch of different pain disorders, including fibromyalgia. And that basic work that was done at the university was ultimately licensed to Pfizer, who took it through clinical trials. And the royalties that Pfizer pays to Northwestern has done a world of good for the university and continued to fuel academic discovery. It accounts for about 20% of the university's total endowment. And they've been able to use this to support, student financial aid. They've built new buildings. They've recruited some of the most talented scientists, Nobel laureates, to work on really important problems. And they've been able to reinvest this money into creating more drugs to help the public. And so our goal really is to do something similar. Imagine if you had an entire institute within a university dedicated to finding new medicines. Imagine what we could ultimately accomplish for the world. And so with that, I just want to thank you for your attention. And I want to thank you for all of your support and for helping us along this journey to try to impact the billions of people worldwide who suffer from neuropsychiatric and neurodegenerative conditions. It is a problem that is of an incredible magnitude. It's difficult to even fathom, but we're doing our best to address this issue as rapidly and as robustly as we possibly can. Thank you.
Well, that's all truly fascinating. Thank you, David, for giving us these insights on the work being done at UC Davis on psychedelics and mental health. And it's time for us now to get into our Q&A segment. Many of you have submitted questions prior to the webinar, so we'll dive into those first. Time permitting, we'll get to those and move to the questions posed during the event as well. So David, our first question, which is a topic you touched on briefly in your presentation, are there mental health, developmental, or physical conditions where treatment with psychedelics is contraindicated?
I would say the most common contraindications are psychosis or a family history of psychosis. But there are other conditions as well. Certain heart conditions can preclude people from using psychedelic drugs. They can increase blood pressure, and in chronic use might actually lead to cardiac valvulopathies. And so there's not only psychological conditions, but there's these physical conditions that can preclude their use.
The next question is, does psychedelic-assisted therapy plan to tackle more serious diseases such as Alzheimer's and schizophrenia, or are some brain diseases too scary to touch? Have you studied using psychedelics for Alzheimer's and dementia?
So there are a few people that are interested in using psychedelics to treat conditions like Alzheimer's, particularly because of their neuroplasticity promoting effects, the ability to to regrow these atrophied neurons. In neurodegenerative conditions, the disease undergoes a couple of different phases. The first phase of the disease is actually synapse loss, and that's when you start to have the cognitive impairments. And then there's a second phase where you get physical death of the neurons. It's believed that if you could intervene early enough when you have synapse loss, but before you have neuronal death, you might actually be able to reverse some of the cognitive impairments and prevent further neuronal death. And so many people are interested in using psychoplastogens to do that. There is a group at Johns Hopkins who is trying to do some pilot studies in patients with Alzheimer's disease using psychedelics. But what I will say is that the vast majority of the field thinks that this is a patient population where psychedelics should not be used. And that is because informed consent is very challenging with those patients, and because they can induce some pretty profound anxiety-inducing experiences. And so that's one of the reasons we've been trying to push these non-hallucinogenic psychoplastogens as treatments for conditions like Alzheimer's disease and schizophrenia.
David, our next question was posed on a couple of different occasions. People wanted to know how we can ensure that our patients do not get addicted to psychedelics.
Yeah, so it's really important to specify what compound you're talking about. So classic psychedelics like psilocybin and LSD are actually not addictive in the traditional sense. So for instance, compounds like cocaine and heroin, if you give them to an animal will self-administer those, indicating that it has addictive potential. Animals won't self-administer LSD or psilocybin. So there's very little risk for developing addiction for those classic psychedelics. Now, for some atypical psychedelics like MDMA and ketamine, they do have some addictive liability. And the best way to mitigate that has really been through in-clinic administration. So the patient is only getting access to one or a couple of doses while under the supervision of the healthcare professionals.
Our next question was posed, will UC Davis be doing research on the effects of microdosing psilocybin? This individual said they've had great success with it to heal their PTSD. Does microdosing really work?
This is a great question. I'm glad you asked it because we were the first to ever do preclinical studies on microdosing to understand mechanistically, can it produce therapeutic effects? And we found that we could get some therapeutic effects. We found that even with sub-hallucinogenic doses, we had an antidepressant and an anxiolytic effect in these preclinical models. Now, the problem with microdosing is that these compounds at the moment are controlled substances. And so getting compounds in the right purity and with the right identity is challenging. And so most people who are attempting microdosing on their own are probably not using the right dose, or they might not even be using the right compound. And so it's been a little challenging to really see in humans if microdosing is effective. The other thing that seems to be clear from our preclinical work and from some other human work from other researchers is that the neuroplasticity effect seems to turn on before the hallucinogenic effects. And so as you start to give higher and higher doses of compounds, you start to get to a level of receptor occupancy where you turn on the plasticity. And if you go a little higher, now you turn on the hallucinations. So titrating in that level of compound is very, very challenging. And the reason it's challenging is because we're all different. We all have different levels of those receptors in our brain. We all have different enzymes in our bodies that metabolize drugs. And so usually what happens is somebody is either using too high of a dose or too low of a dose. And so that's why we have attempted to make the non-hallucinogenic variants, because then you can just take high doses and never have to worry about turning on the hallucinogenic pathway.
Our next question, someone's wondering, what is the difference between SSRI's effects versus psychedelics in terms of what it does to the brain?
So the first thing I want to emphasize is that SSRIs, even though they were discovered over 30 years ago, we still don't understand how they work. And this whole chemical imbalance hypothesis has really been debunked. Now, that doesn't mean that they're not effective for some patients. They clearly are. It's just the mechanism by which they work is not clearly understood. What is known is that SSRIs can produce this cortical neuron growth that we see with psychedelics. They just do so on a different timescale and on a timescale that correlates with their clinical efficacy. So in order to get growth from an SSRI, you need to give the drug daily over the course of weeks to months. But then very, very slowly over time, you'll start to see this growth and you'll start to see the symptoms start to be addressed. Psychedelics seem to do this very rapidly, within 24 hours, and the effects are sustained. Every single antidepressant treatment that we know of, be it a pharmacological treatment like an SSRI or a psychedelic, or a non-pharmacological treatment like electroconvulsive therapy or TMS or exercise or acute sleep deprivation, they all seem to do the same thing, and that is promote the growth of those cortical neurons. The key difference is kind of in the rapidity, the robustness, and the selectivity with which they can do it.
Our next question was, what are some future world implications from your research that you hope to see?
Well, I really hope that the concept of using psychoplastogens to heal damaged neural circuits results in a complete paradigm shift in the way we think about treating brain conditions. I want us to move away from trying to mitigate disease symptoms and trying to heal the underlying damage. Think of it like a broken leg. If you break your leg, you get a cast, and then it heals, and then you can take off the cast, and then you're more or less okay, and you can walk again. That's kind of the way I'm hoping that this research will lead. If we can find ways to selectively heal damaged neural circuits, perhaps we don't need to be taking a drug every single day for the rest of our lives. I also want to emphasize that it is unlikely that we are going to cure any of these diseases. That is a very, very high bar to clear. But that might mean instead of taking a drug every day, maybe we take it once a week, once a month, once a year. That's unclear, and it will be dependent probably on the patient and the disease indication. But that is really a paradigm shift in the way we think about trying to address brain conditions.
Right. Okay, David, it looks like we've got time for one last question. What is the current thinking on whether psychedelic hallucinations are a side effect or therapeutic? Is it possible that the psychoactive effects themselves are actually important therapeutically?
I would say that for certain patients and certain indications, the subjective effects can absolutely have therapeutic benefit, 100%. The problem is the scalability. And so the question is not really a yes or no. There's no yes or no answer to this. What I think the question really is can you get some level of therapeutic effects without the hallucinogenic effects? And if you can, what patients will respond best to that? And what patients will respond best to the hallucinogenic medicines? And so that is why at the Institute, we're developing all of these compounds. I spent a lot of time today talking about the non-hallucinogenic psychoplastogens that we're developing, but we're also developing novel hallucinogenic compounds that don't suffer from some of the other liabilities that psychedelics do, like their cardiotoxicity, like their addictive potential, like their shelf stability, that kind of stuff. And so when I think about treating brain conditions, I always think about a kind of a treatment pyramid. And at the base of the pyramid, you want the most scalable option, the option that is most accessible to the largest number of people. And if somebody doesn't respond to the base treatment, in this case, probably would be something like traditional antidepressant or a non-hallucinogenic psychoplastogen, you start to move up the pyramid. And as you move up the pyramid, you can append additional things to it that maybe are a little less scalable. You can add psychotherapy, for instance, that we know that basically helps anything if you add psychotherapy. But we also know that there's a dearth of qualified therapists for everyone out there. And as you continue to move up the pyramid, I think at the very, very top of the pyramid, that's where you're going to see the psychedelics operating, because the in-clinic administration definitely does increase the cost and the complexity. And so I think they're going to be reserved for only a select number of patients that really require that type of experience.
My sincerest thanks to Dr. David Olson for taking the time to be with us today, sharing how his research at UC Davis's Institute for Psychedelics and Neurotherapeutics is leading to a paradigm shift in mental health care. As we lead the way for the next generation of psychedelic-related neurotherapeutics, the work being done here at UC Davis is nothing short of extraordinary. If you're interested in learning more about how you can support this life-changing work, please feel free to contact me, Jason Woolman, at the contact information shared on your screen. I'd like to thank you all again for joining us today and for your participation. And we look forward to having you join us at our next Plugged In event.